For those of you who aren't familiar with REGENXBIO, we are a gene therapy company and solely a gene therapy company. And more specifically, we are an AAV gene therapy company that's really based on our proprietary NAV technology. And so we've branded our NAV technology, which really includes over 100 AAV vectors, and most prominently, probably that most people in this room are familiar with, include AAV7, 8, 9, and RH10. So, the really basis of the company is the strong intellectual property portfolio we have around all of these vectors. And I've mentioned, this portfolio includes over 100 vectors, including probably 8, which is the most well-known in terms of treating the retina. It not only includes the vectors themselves, but also sequences that are at least 95% identical to these capsids. In terms of the adoption of the technology, it's been widely adopted. So, we've really given a kind of a free access approach to the technology. There's been a lot of academic centers and as well as industry partners and other industry folks who have used the technology that have really helped to advance this idea of gene therapy. And hopefully it's not so much of a futuristic medicine, but here in the present. In specifics to our technology, who are official licensees, you can see that we have over 20 partnered programs amongst 8, or just recently announced yesterday, 9 licensees. I think the ones that have been most significant in terms of advancing the field of gene therapy have been the results in hemophilia, as well as in a disease called spinal muscular atrophy, where there have been some pretty phenomenal clinical results. So we've moved beyond curing mice and getting into humans, and hopefully the hope here is that we'll see similar results with the retina as well. Our internal lead programs, as you can see, is we're a platform company looking at multiple therapeutic areas, in which the retina is one of them. And our lead program, which I'm going to talk about in the next couple of minutes here, is RGX-314, targeting wet AMD in the eye. So I don't need to talk about the disease to anyone in this room, but it was good to hear multiple presentations talking about the continued unmet need around repetitive injections, increased frequency that results in decreased compliance and ultimately efficacy. And the idea for us is really simple. It's using AAV8, which we think is the best-in-class vector in terms of transduction in the eye, in the retina, and using a gene that encodes for an anti-VEGF Fab with a subretinal administration to basically turn the retina into a factory of producing locally produced anti-VEGF Fab to cut down and hopefully eliminate the need for repetitive injections. So a lot of folks have tried gene therapy for wet AMD before, so what makes this different? Predominantly, I think the vector does make a difference in terms of the capsid that's used. The picture on the top comes from a Science Translational publication with Gene Bennett and Jim Wilson, with Luc Vandenberg as the first author, showing and comparing the transduction efficiency at same doses in non-human primates between AAV2 and AAV8 with the GFP protein. We also think using a monoclonal antibody fragment instead of using SFLT will make a difference in terms of leveraging the standard of care in terms of the transgene that's being used. And the hope here is that we would then encode and be able to produce a protein product or a transgene product that would eliminate the frequent and uncomfortable administration of treatment for wet AMD. A little bit of the preclinical data. So if you take indirect comparisons and you look at just protein levels from aqueous taps in terms of the transgene expression level. So you can see here the REGENX vector at the top using AAV8 in a subretinal administration with an anti-VEGF Fab at similar doses across the board. In comparing these 3 different experiments in non-human primates, you can see that at least the REGENX vector reaches a maximal expression in non-human primates around 5,000 nanograms per ml, which seems to validate in a non-human primate model, at least, that this idea of the capsid driving much higher levels of expression. So we're leveraging that into a phase 1 trial. I won't speak too much about this. Dr. Jeff Heyer will be speaking more in detail about our trial Saturday morning in the wet AMD session. I think here's just a brief overview of the trial design. One thing we are doing is there is a required run-in where patients do need to be injected with an anti-VEGF injection and need to have a documented response on OCT prior to receiving the gene therapy. We're targeting a total of 18 patients in 3 different cohorts, around 6 patients per cohort. So that's it.